eLTER, Integrated European Long-Term Ecosystem, Critical Zone, and Socio-Ecological Research.
Okay. Thank you. I hope you can see my screen. Thank you, Terry, for introducing me and the long title of my presentation already. Just for the recording, maybe I will again spell it out. It's variability of ecosystem processes in Europe from different sources and scale. My name is Christian Poppe, and I'm a PhD student in Germany in the IPG3, which is also called Agrosphere in the Research Center in Jülich. And I also want to thank my contributors, Bibi Nas, Alexandra Belflam, Harjan Henriks Fransen, and Harry Verreken from our institute. I want to go straight away into the scientific background of my work, but I want to keep it all simple and very general today, so not many slides about results just in the end. And the scientific background is that ecosystem processes are important because they indicate, for example, if an ecosystem is coping well or badly with environmental changes. So ecosystem processes are, for example, the carbon uptake by vegetation or the release of water from the vegetation to the atmosphere. And it's not yet so clear how these processes or fluxes will change with ongoing climate change and different aspects of climate change, such as the increase of CO2 or temperature and increasing droughts. But I'll come to that now. This evapotranspiration, that is the water use of an ecosystem, and the carbon uptake are very different across different vegetation types or aridities or hydroclimates. For example, semi-arid or arid ecosystems might have evolved to be more sensible to changes in the water availability, so they will adapt the evapotranspiration, so the water use, to save water more dynamically than other ecosystems. Of course, the atmospheric carbon dioxide concentration will affect the carbon uptake, and changes in precipitation and atmospheric water demand or soil water, particularly looking at droughts, will affect also the water that is released and physiological changes in the vegetation, and therefore also the carbon uptake. Also different aspects such as ecosystem demographic traits, so the age of a forest or other geological factors and soil factors also play a huge role here, but these are not really part of my study. Again, particularly looking at droughts, I think it's very important to quantify the response of these ecosystem processes to drought, especially because several studies now, for example, this by Lehner et al., have pointed out that in Europe, the Palmer drought severity index, as shown here, will increase when comparing a 2 degrees Celsius increased future climate to a present day climate. So we will have in future most likely increased drought frequency and also more severe droughts. And we do not yet fully understand how these ecosystem processes will react to droughts, especially because if we look at these fluxes from different scales, so from plant scale measurements or leaf scale measurements, even to ecosystem measurements or more land surface model, continental scale and grid-based data, there are several signals that might contradict each other. So we're still looking for some robust understanding on this response to those. And also regarding the increasing CO2, will it counteract the changes from the increasing drought severity, or will it even aggravate it? That is still some quite open question. So as I mentioned, there are several data sources from different scales about these data that range from leaf level, plant level, plot level to whole continental level. And here I want to showcase just a difference between those scales. On the left-hand side, I plotted a point for eLTER sites that we have selected in our task in the eLTER Plus project that will offer or have submitted already with the data call data that is valuable for our research. And then the color will indicate just very simplistic based on annual precipitation amounts, if it's a very humid or very arid or something in between hydroclimate. And even though we have a very nice coverage in Central Europe, we don't have good coverage of the Iberian Peninsula, for example, in particular. And this is especially interesting because we are interested to find robust understanding of these semi-arid or arid ecosystems to these environmental conditions. And if we just have 1 or 2 to 3 sites in these regions, it might not be that significant. And on the right-hand side, the map is from such now gridded or raster data set from a model or model data fusion. So I model incorporating observations and already updating the fluxes and states based on the observations and reducing some of the model bias. And due to this methods and techniques, we're able to get this gapless time series and estimates, and look at all the points here in the map and have more robust signals for all the different hydroclimates. But if we now want to compare these 2 different scales of data, we encounter some problems. Here on the left-hand side, I painted in red dot that could be, for example, a eddy covariance tower with the footprint, again, very simplistic, as a red dashed line around it. So this is a forest eddy covariance tower. And if we want to compare it to, let's say, remote sensing data, we might just look at the closest grid cell. I'm not sure if you can see my mouse, but it's the one where the red dot is inside. So indicated by the boundaries of this black dashed lines, and the grid cell center, the black dots. And this closest grid cell is actually a bit misleading now if we compare the data, because it is mostly representing croplands, and just a small share of the grid cell is actually looking at the forest. So there's again different techniques how to do this comparison. You can look at this other grid cells that are around here and that have more dominance or more coverage of that ecosystem that is represented by the eddy covariance tower and so on. Or land surface models nowadays can also give estimates on this sub-grid cell composition. So for this closest grid cell, for example, you might be able to disaggregate the time series to get just the forest fraction of these fluxes. But still a lot of a disagreement on in the time series and uncertainties. So this is one example when I was looking at that using the Wüstebach site that is in Western Germany, close to Jülich. On the left-hand side, you can see the location of the site, and on the right-hand side, one example of the closest grid cell, so to say, from a satellite picture. And this grid cell is 3 kilometers big, because all my studies I base on a 3 kilometer resolution, because I have outputs from a land surface model on 3 kilometer resolution. So to streamline everything, I am using the same resolution. And you can see here that this is a good example, because the grid cell is covered mostly in forest, as far as I can tell at least. And here there's this red cross, which is the site with the footprint around it, is also a forest site. But there might be very different cases, for example, somewhere else. And if I look at the whole ICOS data set, for example, so mostly focusing on eddy covariance towers and their data, we would have almost half of the sites covering evergreen needle-leaf forests, and the rest of the sites will cover croplands, deciduous broadleaf, and grasslands. I extracted some land cover because they are not functionally represented in the land surface model here, CLM5, that I mentioned here in the point. I will introduce you to it in the next slides. So for example, mixed forests don't really have a functional representation in the model, which is also some kind of uncertainty, or not a nice representation, because the functionality of mixed forests, such as increased resilience, for example, is not included in such models yet. So now to this community land model in its 5th version. How is the land surface represented here? So we have first the grid cells, or first you have this whole amount of grid cells, but then you have one grid cell, and then one grid cell is divided into several land units. It might be natural vegetation or crops, amongst others. And then this, for example, the vegetated land unit that is on the lower upper right here, this green fraction, might be divided up again into plant functional types. And these are these computational entities that will determine which sets of equations will be used to compute and simulate the fluxes, and also which sets of parameters. And then these equations will be solved, and they describe surface energy fluxes, hydrology, and here a novelty in the CLM5 is that you can also have resolved biogeochemical cycles. So vertically resolved carbon in the soil layers, or also inside of the plants, you would have the carbon pools inside of each organ, such as the stem, the leaf, and the roots. And that's now getting upscaled inside of the model to the whole grid cell, and then to the whole domain that you're trying to simulate. And for my case, the extent to this blue shape, it's a bit weird. It's not like a rectangle, because we're really trying to keep this consistent 3-kilometer grid cell area. And in the north, it's a bit distorted here in the projection, but all the grid cells inside of this blue domain region of our model will have this 3 times 3 kilometer, so 9 square kilometer grid cell area. And now coming back to how this ICOS data set is represented inside of that European model. Now just looking at the grid cells that correspond to an ICOS site, and looking how the land surface looks in this subset of grid cells. First, looking at this inner pie here, the land units, you can see that 3/4 of the land units, or the land surface, is natural vegetation, which is quite, I think, a good estimate, or a good representation, and 1/4 of it is crop. If we go back just quickly here, you can see that around 1/4, is this green croplands, and the rest is forest or grasslands, which are natural, at least for this use. And then dividing up this natural land unit, as I explained it, into this plant functional types. That's the donut, or the 3/4 donut around it. And looking at this ENF, that is evergreen needle-leaf forest, that before in the ICOS dataset was almost half. Here is just represented by this pink color and this olive green color. which is around just a quarter of the whole land surface, so this is a bit underrepresented. And then we have an overrepresentation of this orange that is the grassland, and also this light brown that is Arctic grass belongs also to it. So that might cause some bias when comparing this data from the continental model with the ICOS data set. And this is oftenly done also with other models because we have to compare to somehow have this ground truthing, because of course the in situ data from the sites should be more accurate and include the variability that we want to simulate also in the model. Okay, now already to some results. I'm comparing different models, but here the red one, the red time series or the red line is the CLM5. And we see more often than not an underestimation by it for the evapotranspiration, so the water use of the ecosystem. And on the upper left diagram, it's aggregated over all the sites and all the corresponding grid cells from the ICOS dataset that correspond to the evergreen needleleaf forest. So here we have an underestimation by CLM5 throughout the year, but other models from remote sensing, for example, here the yellow one, which is the glass dataset, have overestimation. So in that case, we might argue that CLM5 is really not doing so bad, but still we have to look inside the model to discover, for example, what's happening here in autumn, where the underestimation is much larger. And in the lower right, we see in croplands, there all the models, also CLM5, are overestimating in the second half of the year. And this is an important point because also croplands are heavily managed and harvesting times and management practices are not yet included, especially in the large scale in this continental models. So this might cause also huge biases, and here where you after the harvest, for example, CLM5 wouldn't know that there are no crops anymore, and then that's one possible explanation why CLM5 is overestimating in this like late summer or early autumn. Okay, and then aggregating also over this specific seasons, winter and summer here, for example. And on the different land cover here, the different shapes and the colors are again different models. And we see a huge range in winter across the land cover types. So we see that the colors, the different shapes from the same color are more closer together sometimes or more often than the different shapes. And that means that the model is not distinguishing the plant functional types enough, so that you would have a more similarity between time series of the same model, but from different plant functional types, than between different models with the same plant functional type. I hope that's clear, but it means basically that I think still the model structure is not good enough representing the differences between the different land cover and plant functional types. So this is something that we should be working on also in terms of parameter representation or the whole model structure as such. Okay, so that's already it. I'm summarizing now what I wanted to convey in my presentation. It's that quantification of ecosystem processes is important, and the different sources across the scales disagree on the response and the time series because of many reasons. But land surface model can estimate these ecosystem processes in very large scales and which might be of use for us. But we have to still consider the different land surface representation between the land surface model and the sites, or land surface models and the different ecosystem types that we want to distinguish and analyze. Okay, and then the last point is that this discretization of the vegetation functional types does not include important aspects such as mixed forest types, as I mentioned, and human management, and therefore we might have still some huge bias in that data. And thank you now